Sure, thank you so much. And it's really a pleasure to have you, Christina, and everyone that could join us today. We're really excited to be able to present to you. And so our project is called the Pilot Evaluation of Targets in Neurofibromatosis Type 2 through Drug Potency and Viability Analysis by the S prime metric, which we'll explain a little bit later. And we took a computational approach to analyze a drug screen for neurofibromatosis type 2, which was the main goal of this project. And so I will pass this to Mia to kind of go into the biology background. I assume you're already more of an expert than we are, but just kind of as formality's sake, Mia, if you want to take it away.
Yeah, of course. So here we have the genetic basis for the NF2 gene and the myelin protein. It is a rare genetic tumor predisposition syndrome caused by mutations in the NF2 gene. It occurs in approximately 1 in 25,000 individuals worldwide and is characterized by development of vestibular schwannomas, meningiomas, and spinal tumors. Most patients are diagnosed between 15 and 30 years of age, and these tumors can result in hearing loss, balance problems, neurological deficits, and reduced quality of life. Here is some of the current treatments we've researched and looked at. So currently there is no cure, and there are some current treatments that focus on benign tumor removal and symptoms. There is surgical intervention for vestibular schwannomas, meningiomas, and spinal tumors, but this can lead to a risk of nerve damage, and there's also radiation therapy. There is also additional clinical trials that are coming, but because of its extreme rarity, conducting large clinical trials in NF2 is generally unfeasible. Finally, there's emerging gene therapy. There's like ongoing research of delivering functional copies of the NF2 gene via adeno-associated virus vectors to stop tumor growth. The myelin protein loss drives tumor formation, as you must know. The NF2 results from loss of function mutations in the NF2 gene that encodes for myelin. Myelin acts as a tumor suppressor protein, and myelin is involved in cell proliferation, cell survival, cell-to-cell interactions, and the co-localization of proteins, often near cell junctions, which is important for regulatory signaling. The loss of myelin also activates multiple oncogenic signaling pathways. These pathways represent potential therapeutic targets. And then this is like a large-scale diagram that shows multiple complex and interconnected pathways of the myelin protein and the NF2 gene. Next, we have the study design, which I will pass on to Olivia.
Yeah, so I will go into a little bit of what we did with the data. And so we pulled our data from a paper called A Chemical Biology Approach, identified PI3K as a potential therapeutic target for neurofibromatosis type 2. And our data was from their open access NF2 compound screening study. It was a 384-well high throughput screening of 40 candidate compounds. And we looked at their Merlin null and wild type Schwann cell models. The paper mentions the assay having an assay quality of Z prime score of 0.79. And their original goal was to use their chemical biology approach to survey drug candidates for NF2 by exploring their nuclear morphology and viability endpoints. However, for us, we narrowed in on two specific screening endpoints in the study, which was EDU uptake and number of nuclei, EDU being a fluorescent stain targeting DNA synthesis. So we're able to track the cell proliferation, while number of nuclei helps to track cell survival and cell death. So within this data, we kind of took three analysis approaches. So firstly, we were prioritizing individual compounds. So we ranked drugs using S prime, which we will go into a little bit later in the methods, but essentially it's a metric that combines the potency and max effectiveness of each compound. And from that, we use delta S prime, again, which we'll explain a little later, but that is used to identify compounds that were acting more strongly on the test cells than on the wild type reference cells. Our second approach was to compare the viability endpoints between EDU uptake and nuclei count responses. So we were determining whether each drug primarily inhibited cell proliferation or reducing cell survival. And from that, we were able to distinguish cytostatic and cytotoxic effects by comparing their performance across both endpoints. Finally, our third approach was to identify any promising mechanisms and therapeutic pathways. So we grouped their compounds by their MOA to identify any consistently high-performing drug classes. And with further analysis, we connected those mechanisms to signaling pathways affected by myelin loss. And we we're able to prioritize targets that may need further testing or combination therapy. And so I will pass this to Suhan to go into our methods.
Thank you. Now I'm going to talk about our methods. So basically, our goal was to compare how different drugs affected NF2 mutant cells and normal cells. And instead of just looking at one measurement, we used several ways to evaluate each drug. And then this helped us figure out which compounds look most promising for future research. So here we have some dose response curves. So first we use something called a dose response curve, and this basically is like a graph that shows what happens to the cells as the amount of drug increases. And from these curves, we measure things like how strong the drug was and how much of an effect it had. And even if the two drugs have a similar overall effect, one might work over a wider range of doses, and that's what can make it like easier and safer to use. Oh, next slide, please. Okay, now S prime introduction. So basically, we used a score called S prime, and instead of looking at like a lot of like numbers, like several different numbers, basically what S prime did, it combined all of them into like one score, and basically that made it like a lot easier to compare all the drugs. We also calculated the delta S prime, which tells us how differently a drug affects NF2 mutant cells compared to like normal cells. And then we also calculated two S prime scores. One was based on the EDU uptake, which measured cell division, and then the other was based on nuclei count, which measures how many cells survive. Um, then we can go to the next one. And then
Just one,
EDU
one
versus
thing,
nuclei.
Suhan, one thing real quick. Just wanted to
Oh,
make it real clear that if you could go back one slide, Olivia. The basis for the S prime is that zero asymptote minus infinite asymptote. So this is basically the overall maximal effect size you can achieve over the whole concentration range, and that's in a ratio with the EC50, which is a statement on potency. So the
mhm.
fundamental thing inside of the single value logarithmic scale unit is a comparison between effect size to potency. So just want to call that out because we build on that. So
Oh, yeah.
sorry, you Suhan, please continue.
No worries, no worries. Now we're into EDU versus nuclei. So basically, these two measurements tell us very different things. So EDU uptake shows whether the cells are still dividing. And if the EDU goes down, it means that the drug is slowing or stopping cell growth. And the nuclei count tells us how many cells are actually alive. And if the nuclei count goes down, it means that the drug is actively killing cells. And looking at both measurements like together helps us understand exactly how like each drug works. And then now processing, like after collecting all of like our data. We process everything so the results could be compared fairly. So we standardize the data and use the Hill equation to calculate the S prime scores. And if the same drug appeared more than once, we average the results together. We also calculated delta S prime whenever we could compare mutant cells with normal cells. Um, and then finally, we ranked all of the drugs from strongest to weakest, and this basically helped us like choose and identify the best candidates for like future testing.
Okay.
I'm Anushka, and oh, sorry, go ahead.
I was just going to pass it to you. You got it.
Okay, awesome. So I'm gonna go over the results. So the first slide we're looking at summarizes our primary S prime and delta S prime rankings. So to avoid overstating stuff, I'm just gonna briefly go over how we interpret these values. So as Olivia mentioned already, S prime combines potency and maximum effectiveness into one score. And a higher S prime score means stronger drug activity because the drug is effective and works at a lower concentration. And here we've compared our drug responses for EDU and nuclei count. So the S prime value for EDU reflects the effect on DNA synthesis. Meanwhile, for nuclei count, it reflects the effect on remaining cell population. So the way that we calculate delta S prime is wild type S prime minus NF2 mutant S prime. So therefore, a more negative S prime means that the drug inhibits the mutant cells more strongly, and which is what we want. So we can see that our best drug candidates are really the ones that have the most negative delta S prime. A delta S prime near 0 means that there's a similar effect on both mutant and wild type cells, which is doesn't really help. And a positive delta S prime means that it had a stronger effect on wild type cells, which is what we definitely don't want, which is the one drug that has a value of 17. So we just wanted to show you a range. And moving on to our top drug candidates. So the first one we looked at is Genetisbib. It ranked first for EDU and second for nuclei count. It had strongly negative values for both endpoints, suggesting that it has a strong inhibition of proliferation, and it also strongly reduces remaining tumor cell population. So the way that this drug works is by inhibiting HSP90. HSP90 is a heat shock protein, which stabilizes. It's really it's a molecular chaperone which helps proteins fold and remain stable. And by inhibiting HSP90, we kind of disturb this complex that it has with the receptors, leading to a loss of signal in the PI3K and the MEK pathways, which are important pathways for tumor cell proliferation. So without HSP90, the signaling proteins become unstable and become degraded, and which eventually leads in promoting cell death. It has mild to moderate side effects, which are listed below. Moving on to our second drug, which is also actually a heat shock protein HSP90 inhibitor. This AUI-922 ranked second in EDU uptake and first in nuclei count. And this balanced response suggests that it has effects on both proliferation and decreasing cell number. So has more of a cytotoxic response as opposed to cytostatic response. As seen in the figure, as shown in the figure, AUI-90 also destabilizes the HSP90 complex and the IGF-IR beta protein, which results in growth inhibition. And this also targets the same pathways that we talked about earlier, the PI3K and MEK pathways, and leads to ultimate apoptosis. It also has moderate side effects, which are listed below. Moving on to the next drug, which is crizotinib, which ranked third for EDU uptake and only 9th for nuclei count. The stronger EDU ranking suggests that it has a more prominent effect on proliferation than on reducing cell number. So crizotinib inhibits specific receptor tyrosine kinases, specifically the HGFR receptor, which is the hepatocyte growth factor receptor. Not entirely sure if I'm pronouncing that right. But these receptors sit on the cell membrane and transmit growth signals, which leads to a signaling cascade. So once activated, they also target the PI3K and MEK pathways. And crizotinib works upstream compared to the drugs that we just talked about. It inhibits the receptor itself, so it blocks the signal at the top of the pathway, which reduces downstream gene expression, ultimately leading to apoptosis. It has also moderate side effects, and which are listed below. Moving on to our last drug is XL765. It ranked 7th in EDU uptake and 3rd for nuclei count. Oh no, you're good. Sorry. It ranked 3rd for nuclei count, and the delta S prime for nuclei count is considerably more negative than its EDU delta S prime, which suggests that it has a stronger effect on cell population than on DNA synthesis alone. So XL765 is a dual PI3K and mTOR inhibitor. PI3K is a really important pathway for cell survival and growth, and mTOR also regulates protein production, cell growth, and survival. So XL765 blocks the pathway at multiple points, which reduces downstream signaling and gene expression needed for tumor growth. It's in phase 2 of clinical testing currently and has moderate side effects, which are listed below. Handing it off to Olivia, who's going to take a look again at our primary S prime and delta S prime rankings.
Yeah, so after reviewing the drugs, we're kind of coming back to this chart from previously on the S prime and delta S prime for each of these drugs. So a couple things that we were noting from this list was, firstly, we have Genetisbib, which had a slight EDU bias in their delta S prime ranking and had the highest magnitude of mutant versus wild type differential drug response as seen in the endpoint difference here. AUY 922 also had a good magnitude response, but didn't have much of a major endpoint difference here. Same with crizotinib, it had moderately good delta S primes for both, but not much of an endpoint difference. For XL765, so there was some EDU selective drug response, but we found a much stronger nucleotide count endpoint with this endpoint difference of 2.2. And finally, this last drug, MVP BGT226, was included as a contrasting compound to show a strong EDU endpoint with wild type preference. So moving into our next analysis method, we were comparing MOA between the EDU and nucleotide count endpoints for the mutant cell line. And so we created this chart of difference between drugs that were skewed by nucleotide count versus by EDU for their S prime values. So to make this chart, we calculated S prime values by percent EDU and number of nuclei for all targets from the NF2 data set. And we collapsed this data set to calculate median values for all targets that had multiple occurrences across. And from that, we calculated the difference between EDU and nuclei and created this bar chart that visualized targets skewed by nucleotide count versus by EDU uptake. And just as a filtration note, only targets with repeats of NFs greater than or equal to 2 were included. And we noticed here that most of our targets were skewed by nuclei count, which we will go into a little bit more interpretation in our discussion. But for now, just a couple notes from this chart. We noticed that
Olivia, could you restart over? Your mic was doing a fade in, fade out.
Oh, sure. Is it okay now?
Yeah, it's okay right now.
Okay. Did you see the last slide?
We're good on the last slide, just the start of
Okay.
this slide.
Sure. So we noticed that a large section of were cell surface receptors. So in this graph, 48.6% are cell surface receptors, and all of those are tyrosine kinase receptors, except for HDH1 and OPRD/DOR1, which are G protein coupled receptors. And also, only one of 35 of these were related to mitosis or the cell cycle, which was CDK1. So a little bit of some takeaways from that, where we noticed 44 out of 48 MOA categories had a higher nuclei count derived S prime than EDU derived S prime, noting that generally there is a trend where nuclei count derived S prime is observed as the higher magnitude endpoint across both the mutant and the test cell lines. And then underlying that, we also noticed the mutant cell line had a higher nuclei count endpoint skew compared to the reference. This difference is most dominantly expressed in the cell surface receptor related MOA classes. And now I will pass this to Aria to finish up our key conclusions.
So to summarize our primary findings, HSP90 inhibition was our top performer across both endpoints, with Genetisbib and AUY 922 consistently ranking at the top. Interestingly, PI3K inhibitors yielded lower S prime scores than we originally hypothesized. By comparing the divergence between EDU and nuclei responses, we were able to pinpoint compounds with purely cytostatic activity versus those driving cell loss or cytotoxic activity. This divergence highlighted NF2 signaling redundancy, specifically showing how cells adapt to single-agent treatments. This strongly suggests that combination therapies, like pairing PI3K blockers with drugs that target secondary pathways, will be key to overcoming therapeutic resistance. So, evaluating our framework, a major strength of the study is that we didn't rely on a single readout. Combining EDU and nuclei endpoints allowed us to cleanly separate cytostatic from cytotoxic across a high-throughput dataset aggregated by mechanism of action. However, we have to acknowledge a few limitations. Our dataset had limited delta S prime representation for baseline healthy comparisons, and standard in vitro screens don't fully capture complex tissue physiology. Additionally, because this dataset relies on mouse Schwann cells, further validation is required to confirm that these exact biological mechanisms hold true in human models. Moving forward, a future goal is to validate these findings on the bench by testing our top-ranked HSP90 and PI3 candidates in direct lab assays. To bypass the resistance mechanisms we'd identified, it is important to test combination therapies, specifically combining PI3K inhibitors with FAK inhibitors to block the cell's escape loop. Beyond the bench, it is necessary to transition from mouse models to human NF2 deficient cell lines to ensure selective toxicity carries over. Finally, future work should refine the S prime algorithm itself by including larger, broader datasets with expanded delta S prime metrics to eliminate cell line biases and improve our predictions.
All right, and that's pretty much all we have for you. Really would love to continue the conversation. So we kind of just wanted to ask if you have any feedback, does our approach make sense? And if you have any concerns or improvements from our approach, and then also if you think there's any future work or data that would make sense for us to look at if we expand into a broader set.
I can jump in very quickly, but I know this is Christina's area because this is all the work that she's generated in the data and the cells. But I just wanted to say this was a very thorough presentation. You guys really understood what you were doing and why you were doing it. And I really love seeing, one of my questions was next steps. What are you going to do next with this? But it looks like you really thought out what could be done with this and how there could be future impact. So this is wonderful. Let me see what Christina might ask because she knows this information inside out.
Okay, thank you. And I agree with Irene. The students, Olivia, Anusha, Suhan, who am I missing? Was it Ulysses? No. You've all done an excellent job. This is really impressive for high schoolers. So you're on a level with college students. So you need to be really proud of yourselves. Ari, I don't know if I mentioned your name, Mia. So I guess I was confused in the very beginning because you start your presentation saying that this was reported in the chemical biology publication. And unless I added the CENIX data that you analyzed as a supplemental, this whole chemical approach was looking at a different library of 2,000 compounds showing that this approach was feasible. But you talked about the 40 compounds, right? This is a 40 compound data set you looked at?
Yes.
Okay, so that was the CENIX project. And that's when the company CENIX in Dresden did the ED incorporation and the nuclear counts, which were very powerful way to analyze total cell number just using phase microscopy at specific endpoints. So I guess at the beginning I was like trying to remember, and I remember that we didn't report that as the prime data set in this paper. But your HSP90, you use different ways to analyze the data, and you got prioritized the same targets that the field has prioritized. Actually, HSP90 was back in 2013 a protein of interest, and I think even a company was started focusing on HSP90 for NF2. And I guess at that time, the compounds weren't very advanced, and it didn't continue. Crizotinib has been in clinic in patients. So you did an excellent job understanding the mathematics behind your calculations, and understanding mechanism of actions. And yes, you found mostly RTKs because the group at, I think Annette Bacher had just started with CTF. She and Marcanievo and others had come up with the list of compounds. And at that time, tyrosine kinase inhibitors were were inhibitor class that was very much of interest to cancer biology in general. We've gone on to look at using different assays that I'm not sure if Irene would and CTF would allow us to share, which is using high content imaging over time. And that would give your group a different kind of data set to be able to examine and maybe even train some AI models in looking at morphological differences. Because one of the things we've noticed with some of the drugs we've looked at for CTF and for other projects is that you can have very pronounced changes in morphology, and you might have small differences in total cell count. So some MEK inhibitors cause cells to flatten. So it looks like the cells are growing or that the cells are proliferating. They're really not proliferating, they're just getting bigger. So if you're looking at confluence measurements, you think that the cells grew, but they didn't, they just spread. Other drugs cause vacuoles to form inside the cells. And once you get to understand the morphology of your cell, you can just look at a cell that's been treated with a certain class of drug and say it's responding. And there might be some cytoskeletal changes going on that would indicate perhaps a different target. And yes, you were right about the mouse Schwann cells. Those were the first models we had. Since then, we have patient-derived human schwannoma cells and other human schwannoma model cells. And in some respects, with the cytotoxicity, we see a difference between human and mouse schwannoma cells. The mouse are not as resistant. If you cause a cell cycle arrest, they're more susceptible to undergoing some kind of cell death routine. Whereas the human schwannoma cells, when they undergo cell cycle arrest, survive. They don't undergo cell cycle, they don't undergo, let's say, apoptosis very easily, caspase-dependent apoptosis. They kind of just sit there and rewire themselves and they wait. So there are some differences between human and mouse Schwann cells. Do you have any specific questions for me?
Well, one of the things that we would love to do, as we note that the students are now in transition, the former seniors are moving on to college and the juniors in the program are becoming seniors. For us, moving forward, it would be wonderful if we could figure out some way to be able to work with new test data. If that would be even remotely possible, that would benefit, the students, and perhaps it might even benefit the organizations and people that were supplying the data. It would certainly be from a mentor perspective, it would be a wonderful thing to be able to look at. And
I can put a quick link in the chat. I think we have data. I think that remind me, Ulysses, in the beginning of this project, this came from the NF data portal. It was part of the hackathon. Is that how this?
That's correct.
Came about, right? This is part of the hackathon.
That's correct.
And that Salvo was part of, and you used portal data. And I don't think at that point the synodose for NF2 schwannomatosis was on the portal, or was it? I don't recall. But we do have quite a few sets of NF2 related schwannomatosis data sets on the NF data portal. They're available for use, and we want people to reuse them actually, and gain new insights. And if you can share with us, we'd be more than happy. I would encourage you to look at the synodose for NF2 data sets. The other place that we've combined data is Pluto Bio. It's a user-friendly interface to visualize data, and we've pulled key data sets from the portal to Pluto Bio. And so I don't know the composure of this group. I assume some of you are biologists and some of you are data informaticians. It's a really nice mix. I can hear in the presentation. But Pluto is designed to look at data in a very simple way for the non-bioinformatician like myself. I'm not a bioinformatician, but I love to look across data. So I can just put a few links, and you are welcome to ask questions about the data sets, but there is data to be reused. And Christina, you referred to the phenotyping data. Is there, I was looking at synodose, we don't have any imaging. Is there a data set that you were thinking about?
Well, I know that we capture images for like our current project with CTF, but we don't download and ask Sage to store it.
I see.
But that's something that can be done. The data set that would be great for combination for the next step is the synodose 2, the MIPES combination library that was done together with Mark Ferrer at NIH and CATS. They did the MIPES library, and they ran 2 sets of every drug against every drug in I think 6 doses, and then later on in 10 doses. The advanced combinations got done in 10 doses, and they did both a caspase dependent assay and a cell titer glow assay. And so that NCAT's combination drug study that led to brigatinib is available through this at the Synodus portal through Sage Synapse, I think they call it. And then we'd have to talk, Irene, as to whether it's worthwhile sending high content images, because we often capture green object count looking at as a second measure for cytotoxic drugs. And then we capture the phase every 4 hours for 3 days. So it's a huge data set that takes forever to download, but we've done it in the past. Whenever we got to clear our servers, we're like, okay, let's put it on a secondary hard drive so we have it archived.
It would be nice if we could access that, but I need to point out that one of the focuses that we have in the student program has to do with focusing on dose response curves. So if the imaging data could be transformed or is already transformed into dose response data, that would certainly be ideal, because that would allow us to bring a strength to the analysis and do it in a way that could give an alternative to the current ways of looking at things, which are specifically the area under the curve or EC50 comparisons, which we think to be somewhat, could be improved on, put it that way, for certain applications.
Yeah, it sounds like it's a third dimension almost, or, you know, third data point to incorporate. But it is a ton of data. So I think, as Christina mentioned, figuring out how to, not storage, actually data transfer, right? So where do we store it and transfer? That's one of the difficulties with imaging data, is we don't have a great way to store and transfer it. And then depending on how you're going to phenotype and analyze it, you would need a processor to download it on.
Yeah.
So right, because you're going to have to take it off the server or off the portal and put it elsewhere. So the data is there. I think the logistics are difficult.
Let's follow up on that. We do have with the nonprofit some data processing servers, and they have large data storage and compute capabilities. And we do absolutely have people, not necessarily as students, we'd probably bring a mentor on, but that can do some of the AI and the image processing. We do have those within our nonprofit generally. So we could look into that. I'll follow up with Ulysses, and we'll reach out when we have a little bit of opinion on that.
And then just to understand the problem there is methodology for the images seems to be okay. It's just, it's all related to data. Am I understanding correctly? Like storage, sending, retrieval, so on and so forth.
Well, when you say methodologies, I don't think we have a clear phenotype. I mean, it would be really exploratory work, right? Like to see how, Christina, I don't think there's a clear, like, high content image phenotype that has been validated, right?
Sounds like a cohort 3 project to me.
Right. And Paul, you were saying the data, these images actually provide the data of which we do the drug response curve. So there are dose response curves. And basically a dose response curve comes over well confluence. These are 384 well plates. And then you just, track confluence. You start at about 25 to 30% confluence in that area, and you track it over time. So you do generate, and that's what we send very easily, are all the raw data, the normalized data, and then AUC curves. But then we're looking at the images and we're like, there's so much data here that we don't know what to do with. We're like, oh, this drug makes them, all these vacuoles appear. Some drugs, it's interesting, and all that, more granular data is just not being looked at. It's there. But for instance, we're downloading sets of images and we're occupying 5 different computers and downloading, and we're slowing the actual processing of people working with the one program. So we have to oh no, we can't do 6 at a time. Maybe we can do 4 at a time. And then it's like we were dealing with 400 gigabytes of data, and then IT gets very, very peculiar about how we're sending the data. So, but it's just a process. You go through it, you understand which is the best way to send the data. And I mean, we're even putting it on hard drives and mailing a hard drive. But then it's like data corruption, etc.
We would want to make sure that, if we did engage, that it would be something that would be a benefit to the students, that would be a benefit to you guys, so that you get something out of it for the time and all the background work that you've already done. So it, if we can come to alignment there, any way that we can work with you, I think we would be open to do that, especially if we could get buy-in from some of the students relatively early on, so that they know what's going on and can get incorporated into the project very early. And that would help them. It would also help the program development as we sort of move along, because we would be incorporating their points of view and concerns and difficulties in translating the biological data into their understanding level, which we will have to be able to do as well.
So you're all AP biology students, or how did this program?
Actually, Olivia, I think probably can speak to most of that.
Yeah, so a couple years ago, we started a club at our school that was called the Biomedical Club, essentially kind of helping students to get like pre-professional opportunities and that sort of thing. But one of the things that we organize every year is called the Biomedical Exhibit, and we bring in professionals from local organizations to present about the work they do and kind of just help students on that track and give advice. And so at our very first one, we met Matthew and Paul and the DMV Petri dish, and we got introduced to them. And then from there, we kind of started working with them on NF1 data and now NF2 data. But most of our students are a part of that club.
And they commit for like 1 year and then they graduate. Is that how it works? Or is it a 2-year? What is the?
It kind of just depends on when they join. So I joined as, I believe, a junior, and I've just been doing it ever since then. Our previous group was mostly seniors, so they've already graduated. But just, I guess, when people join, they kind of join for most of their high school time or until they decide they don't want to do the project anymore.
Yeah, so we'll call it 1 year with optional renewal.
Exactly.
And it's something you don't do for credit. You just do for experience, put it in your resume. Okay.
Yeah,
Like
I just wanted to add.
Oh, go ahead.
Sorry. Oh, I just wanted to add really quick because Christina asked if we were AP Bio students or not. There are a couple of us that have taken like AP Bio and AP Chem, but I think it is pretty important to all of us that it's not necessary for students to take that course. It's helpful to have that information, but especially this year, we've been recording all our sessions and we've been making videos so that future students who do join, even if they don't have as good of a biology background, they can still learn and they can still definitely benefit from having the opportunity to work with all these amazing people and work on all these interesting things.
Yeah, exactly. We mainly just want this to be like a learning experience for students.
Mhm.
And how long the work that you presented? How long did that take to actually, do all the calculations and put all this together? What was that like?
I think that's been around a year or so. I believe we started last summer, and we've been working through the year until now, on this specific project.
Okay.
You should comment on the separation between the computer components and then the biology components. What was the balance there?
Yeah, so a lot of our work is done by, we really only have one main expert in computer processing. He's actually not here today, but he does a lot of the data processing. He gets us the data back with the calculated values and everything. And then most of the rest of the team is more focused on biology aspects. So they take the data, we kind of filter it down to make it more manageable. And then just whatever results we have, I guess, just researching the compounds, making sure they make sense, creating the figures in development, and also just like general background pathway research, literature review, all of that based on whatever data we receive from Aaron, who is our computational person.
But also at the early part of the year when we were kind of not in the deep of analysis, we kind of took a step back and we're like teaching programming and some basic kind of tactical technique. So we covered like introduction to cancer biology topics, but also introduction to programming and data analysis topics. And that was kind of like a lot of our early work was actually foundational on that, mostly for the purpose of recordings, mostly for the benefit of future cohorts. And it was towards halfway through the year, the latter of the year, that we started to really specialize on the CENIX data and go deeper with it.
It's really neat. Sounds like a great program. We well, first of all, take a look at the data sets that I put into the link. There are we now have 3 different ways of looking at data between Data Portal, Pluto, and I forgot there's also data we put onto CBio Portal, which could be interesting as well. That's more clinical data. So take a look, and then we can always chat again. And then this the high content imaging, I think that's really interesting, especially if the data is sitting there, nobody's doing anything. I hate to waste data, so it's sitting there. We can kind of brainstorm.
Well, and we're a big believer in the morphology. You know, one of the coolest parts of the NF2 was seeing it positioned as a co-locator near the apical surface, and how it was recruiting and involving a lot with the microtubules and that environment near the receptors, and how you're co-locating receptors and complexes near, you know, there's a huge structural dimension to what Merlin is actually doing. And, you know, we were kind of winking at that with the nuclear count data, but the imaging data would have way more meaningful interpretation in it. So we'd be very interested in looking at that just from a scientific perspective.
Yeah, so we can talk about how to share data. I could just share a few images one day or by email, and you can say, this is something we could do or not do. But that kind of image analysis using AI, I mean, that's next generation, I mean, it's current, what's being done now. So, yeah, because you can generate thousands of these images. And if you're doing a big drug screen, you're like, okay, after a while, everything looks the same unless it's really, really different. But I think it's great, and I'm happy to help support this program any way I can. I think Irene, you know, you're the same. So yeah, we're just happy that someone's taking interest on NF2.
Absolutely. Thank you.
Yeah. Yeah, no, and thank you both for being able to make the time to be able to hear a student's presentation. You know, it's for us, it's important that they do this, and that you know, they have the chance to present in front of a audience that you know, would understand and be able to provide good, valuable feedback for their work. And it's also, you know, more just getting it out from the meetings and out there into the world and these sorts of things. So, you know, thank you again. Thank you both seriously for making the time. I know we all have crazy schedules to work with and so on and so forth. So it's definitely appreciated.
No, thanks. Amazing work. I was just going to think, you're in Maryland. It would have been great if you came to our DC meeting last year. But now, you know, we were in Denver, we're in Chicago, but something like this, you can easily present at our conference. We've had actually in DC, I think we had some high school presentations. I don't think this group was there, right? I can't remember now. Maybe.
Did we send anybody to DC, Paul and Matt? I don't think we did.
No, it didn't work out for DC. We were at CENIX, but we hadn't done the DC one.
Yeah. Matt was at CENIX, and in DC, I can't remember what had happened. I can't remember if there was travel that happened during the time. I don't remember exactly the details, but
that would be a good place to share something like this, if it's ever possible. Next one is in Chicago.
oh,
Well,
okay.
Yep.
Early
Yeah,
June, a little earlier than usual.
And it's earlier, yeah, it's early June by a week. I think it's first or second week of June next time. So just keep it in mind. It would be interesting.
Yeah.
So.
Okay, yeah, I think that's definitely something to touch base about. I think, you know, especially if there's a way that we can get, you know, some support for our students to make their way over and so on and so forth. So
Yep.
I think, yeah, I think that could be interesting because our, at least at this point, the way we're kind of molding the program now is either they do a presentation such as this in front of an audience that is relevant, or they do a presentation in a poster format, also preferably in front of an audience that is relevant. So
Yeah, you get to meet the patients, and maybe you know, we can recruit you to the NF2 world. Maybe you'll be the next, you know, doctors and researchers studying NF2 shock, because that's the conference where you meet all the, you know, researchers, the clinicians, and you know, if you're there for the summit, you meet the patients as well. So it's a good place.
And actually, since we have a good chunk of the cohort here, it'd actually be interesting, like, you know, if the students here, you know, who are here as part of this presentation, would, do you think any of you would have interest in, you know, continuing to do NF2 research or something similar, like later down the line? Yeah.
I think so, definitely. I'm actually looking for college labs right now as I'm going to college. So it's been interesting kind of exploring any fields that I can get into. But yeah, definitely, I think would be an interesting pathway to continue.
me too. I mean, I'm obviously gonna say yes. Me personally, I'm really interested in gene editing and CRISPR, and I've worked a little bit on that. But same as Olivia, I'm pretty much taking whatever opportunity the science world throws at me. So I think I'm really lucky to be here at this meeting and just network, which will be useful, I'm sure.
Great. That's what we like.
So see, we
Okay,
have a
so
couple already.
Yeah. we'll be in touch, send an email, and I can work with Irene and see what we can do, or however. Yeah, I'm all for supporting next generation. We need some good minds that can think critically and yeah, want to learn.
Yeah, especially with the rise of AI, right? That's more important than ever.
Yeah, absolutely.
So what we'll do is we'll send you guys the presentation, and then I'll follow up with the student cohort and also Ulysses, and we'll most likely want those, like, at least the preview images so we can start to think about them. But we'll follow up on that topic in the next week or so. So, but thank you so much for your time. I really appreciate it.
Yeah,
We do
and
very
then
much appreciate it.
yeah, absolutely. And then we'll also reach out as well for some of the other things, the behind the scenes stuff, and seeing the kind of the debrief of how the program works, as well as ways to, develop it and make sure it can get even better over time.
Wonderful. Thanks. Thank
Thank
you.
you.
Thank you so much.
Yeah,
Thank
no,
you
thank
so
you.
much.
Great job,
Bye.
everyone.
All
See ya.
right.
Yeah,
Bye.
great job, everyone. Thank you.
Oh.
Okay, the fast